The screen is filled with the high-definition, ghostly stillness of the Barreleye fish, hovering motionless in the crushing blackness. Look closely at this face. At first glance, the anatomy seems recognizable, perhaps even melancholic. You see a frowning mouth and two small, dark eyes staring vaguely forward into the abyss. But your brain is actively deceiving you. Those dark spots above the mouth are not eyes at all. They are olfactory capsules, the fish's nostrils. The creature's actual eyes are the massive, glowing green spheres buried deep inside the transparent shield of its own forehead. This is Macropinna microstoma, the Barreleye fish. It possesses one of the most sophisticated alien visual systems ever evolved on Earth. Close up on the fluid-filled dome, showing the green lenses reflecting the ROV's lights. For over 50 years, science only knew this creature as a mangled specimen. Its most defining feature, shattered by the violence of trawling nets. We assumed it was just another oddity of the bathypelagic zone. But when we finally witnessed it alive in its natural habitat, the perspective shifted entirely. We realized we were not looking at a monster, but a living, biological cockpit designed for a world where like is the ultimate currency. Now, let's dive into the data and uncover the reality hidden in the deep. To understand the Barreleye, we must first understand the paradox of its home. It inhabits the mesopelagic zone, roughly 600 to 800 meters down. Oceanographers call this the Twilight Zone. It is too deep for photosynthesis to function, yet not deep enough for absolute darkness. In this realm, invisibility is the only defense. For nearly 70 years, the true nature of Macropinna microstoma remained a mystery, obscured by the very tools we use to study it. First described in 1939, early specimens were dragged up in deep-sea trawling nets by pioneering researchers. Always destroyed the animal's most defining feature. Taxonomists were left staring at a strange, blunt-headed fish with peculiar, upward-pointing eyes. But they had no concept of the protective canopy that once housed them. The paradigm shifted in the early 2000s, specifically through the work of the Monterey Bay Aquarium Research Institute. Using remotely operated vehicles, or ROVs, like the Ventana and Tiburon, researchers were finally able to observe the creature in situ. But even with advanced robotics, this fish is a specter. Out of thousands of deep-sea distinct dives logged by MBI, Macropinna has been encountered fewer than a few dozen times. It is solitary, silent, and incredibly still. For decades, we studied a ghost, unaware that the true marvel of its anatomy had been erased by our own hands. When the ROV cameras finally walked onto a healthy specimen, the pilots and biologists saw something that defied immediate explanation. The fish wasn't just blunt-headed. The top of its head was a crystal-clear, fluid-filled dome, revealing the internal machinery of its skull like a biological submersible. Inside that dome, suspended in transparent fluid, were the glowing green lenses. The fish was not swimming actively. It was hanging in the water. It was not searching the water ahead. It was staring directly, unblinkingly, upward. It was watching the shadows. But this raised a critical physiological question. If your eyes are permanently fixed upwards to spot prey silhouetted against the surface, how do you eat what you catch? How do you navigate? Or so we thought. To understand the Barreleye, you must stop thinking of it as a fish. You must think of it as an autonomous, deep-sea observatory. The defining feature of Macropinna microstoma, those tubular eyes, represents a radical departure from standard vertebrate evolution. Most fish possess spherical lenses that protrude from the sides of the head, and granting them a wide field of view to detect predators approaching from the flank. But spherical eyes have a flaw. They sacrifice resolution and depth perception for coverage. In the bathypelagic zone, where photons are scarce, a wide field of view is useless if the image is too dim to process. The Barreleye solved this physics problem by abandoning the sphere entirely and evolving the cylinder. The eye structure is tubular, resembling a pair of high-powered binoculars or telescope barrels. This shape allows for a massive increase in the number of rod cells, of photoreceptors responsible for low-light vision, packed into a concentrated area. It creates a tunnel vision effect, sacrificing peripheral awareness for extreme sensitivity and high resolution in a single direction. These eyes are not designed to see everything. They are designed to see one thing with absolute clarity: narrative. However, the architecture goes deeper than just shape. We must analyze the color. The lenses are bright, vivid green. This is not an aesthetic choice. It is a sophisticated optical filter. In the deep ocean, two types of light exist: the faint, blue remnants of sunlight filtering down from the surface, and the blue-green flashes of bioluminescence produced by deep-sea creatures. The specific yellow pigment in the lens acts as a cutoff filter, absorbing the ambient blue sunlight while allowing this filtration system increases contrast. It effectively deletes the background noise of the ocean's ambient light, making the bioluminescent glow of jellies and crustaceans pop out with startling clarity. It is the biological equivalent of a sniper using thermal imaging to spot a target for a smoke. The ROV footage zooms in on the transparent shield. The fish rotates slightly, and suddenly, the green cylinders inside the head move. But for decades, this design presented a fatal logical flaw. If the eyes are permanently fixed in an upward gaze, locked like telescopes mounted to a ceiling, how does the fish see what it is eating? If it lunges at prey, it would essentially be flying blind the moment it broke its upward stare. Evolution rarely tolerates such inefficiency. Biologists debated this for years. Some theorized the fish relied entirely on its olfactory organs, those dark nostrils above the mouth, to guide the final strike. Others assumed it simply thrashed blindly once it was close enough. Both theories were wrong. The 2009 study by Bruce Robeson and Kim Reisenbichler revealed the elegant, transformative truth. When the Barreleye transitions from hunting mode to feeding mode strength, this brings the focus directly in front of the mouth. It transforms the fish's vision from a passive into an active.
Ensuring a high capture rate in an environment where calories are incredibly rare. It is a dual-mode optical system, shifting from search to track in a fraction of a second.
This brings us to the canopy, the transparent shield that makes this species iconic. Why encase this entire mechanism in a fluid-filled bubble? The shield is not merely a helmet. It is an optical component necessary for the rotation to work. If the tubular eyes were exposed directly to the open ocean, the drag of the water would make rapid rotation difficult. Furthermore, the delicate corneal tissue would be exposed to stinging cells and parasites. The shield creates a localized, static environment, a clean room for the sensitive equipment. The fluid inside the shield is crystal clear and, critically, possesses the same refractive index as seawater. Because the fluid inside the dome matches the density of the ocean outside, light does not bend or distort as it passes through the shield boundary. This refractive matching renders the shield optically invisible. If the fluid were more or less dense than the seawater, the shield would act like a funhouse mirror, warping the fish's view of the world and ruining the precision of its tubular eyes. Instead, the shield disappears. It allows the eyes to rotate freely in a lubricated, protected chamber without any optical distortion. This structure is essentially the cockpit of a fighter jet, a transparent, pressurized canopy, protecting the pilot and the avionics from the harsh external airstream while offering an unobstructed view of the dogfight. But what exactly is it dogfighting against? The complexity of this engineering, the rotating tubes, the specific pigment filters, the refractive fluid shield, is expensive. Biologically speaking, growing and maintaining this head takes massive energy resources. In the deep sea, you do not evolve expensive tools unless you are solving an expensive problem. The Barreleye isn't just looking for random scraps. It is built to rob one of the most dangerous creatures in the ocean. To understand the why of the shield, we must look at the Barreleye's diet. Gut content analysis of early specimens revealed a strange cocktail of zooplankton, specifically copepods and jellies. But closer inspection of the stomach contents in recent years pointed to a specific, high-risk food source: siphonophores. Siphonophores are not single animals. They are drifting colonies of specialized clones, essentially massive, living drift nets. Some, like the Portuguese man-o'-war or the deep-sea apolemia, trail curtains of tentacles that can stretch for meters. These tentacles are armed with nematocysts, microscopic, harpoon-like stinging cells loaded with neurotoxins. They are designed to capture the very same small prey that the Barreleye eats. The Barreleye is a kleptoparasite. It steals food from the traps of others. The tubular eyes allow the fish to spot the faint, bioluminescent glow of the siphonophore colony from below. This is a high-stakes heist. To steal the food, the Barreleye must swim directly into the electric fence of the siphonophore's stinging curtain. It must surgically remove the prey from the tentacles without becoming prey itself. This is the true purpose of the transparent head. It is a riot shield. If the Barreleye's eyes were exposed, a single brush against a siphonophore tentacle would be catastrophic. The stinging cells would fire directly into the cornea, blinding the fish instantly. The transparent shield acts as an impervious barrier. The nematocysts fire harmlessly into the outer membrane of the dome, while the delicate eyes inside continue to track the target, completely safe within their fluid sanctuary. This explains the maneuverability paradox. The Barreleye's large, flat fins allow it to hover with extreme precision, inching through the minefield of tentacles. When it spots a trapped copepod, it rotates its eyes forward, locks onto the morsel, and nips it out of the stinging net. The visual shifts to a wider angle of the deep ecosystem, showing the interconnected web of predator and prey. The Barreleye is not just a passive observer of the deep. It is a specialized thief, evolved to its point. This relationship recontextualizes the entire animal. It is not a sad or confused-looking fish. It is a master of infiltration. The sad expression is merely the streamlined architecture of the jaw. The ghostly stillness is the discipline of the ambush. And that transparent, alien head is the armor that allows it to survive in a stinging forest, where other fish would perish.